Now we are going to discuss about the meiosis. First thing is the term meiosis was given by Farmer and Murray. Now the second thing is the meiosis process produces four cells. The meiosis includes two divisions. The first one is meiosis first and the second one is meiosis second. In meiosis first, there is a reductional division, means it leads to reduction in chromosome numbers. So if there is 46 chromosome in the cell, so after meiosis first, there will be 23 chromosome in each cell, and the meiosis first produces two cells. So now we are going to discuss about the first thing is meiosis first. As we know, the cell divides in two stages. The first stage is karyokinesis, and the second stage is cytokinesis. In karyokinesis, there will be nuclear division, and in cytokinesis, there will be cytoplasmic division. First thing is karyokinesis. Karyokinesis is further divided into four substages. First one is prophase, that is meiosis first. So the process that is prophase first. The process prophase first is further divided into five substages. That is lactotin. Second one is zygotin. Third one is pectin. Fourth one is diplotin. And the fifth one is dikinesis. Now we are going to discuss about the first process, that is lactotin. In lactotin, chromatin threads condense to form chromosomes. In this stage, the chromosomes are longest and thinnest. All chromosomes remain in nucleus and they directed toward the centriole. That is centriole. They directed towards the centriole. So they form mainly a bouquet-like structure. That is bouquet-like structure. This shape seems like this. All the chromatids are directed toward the centriole. That is also called bouquet stage. Now the second process is zygoting. In zygoting, pairing of homologous chromosome occurs, that are homologous chromosomes, and the pair of homologous chromosomes are called bivalents. So they are bivalents. In bivalents, there develops a structure in between homologous chromosome which is termed as synaptonemal complex. So that is a synaptonemal complex. Now the third stage is pectin. So in pectin stage, there will be a crossing over occurs in this stage. Both of the chromatids of each chromosome becomes visible. So both chromatids are becomes visible. So each chromosome are called the tetrad. The crossing over occurs between the non-sister chromatids. Non-sister chromatids are the chromatid that is homologous chromosome. They both are called the sister chromatid. The chromatids are the homologous chromosome are called sister chromatid. Non-sister chromatids of homologous pair develops recombination nodule and exchange their parts, so that is called the crossing over. So the variation that is present in offspring is due to crossing over that occurs in Pachytene stage. Now the next stage is diploidine. So the synaptonemal complex that is formed is begun to dissolve, and the chiasmata becomes visible where the chromatids are attested. This part of the chromosome is called the chiasma. Now the other stage is, the last stage is dikinesis. In diploidine stage, the terminalization becomes start. So the terminalization begin to start, and each chromosome separates, and the nucleolus and the nuclear membrane becomes disappear, and the centriole move towards the opposite poles. Now that is the prophase first stage. Now the second stage is metaphase stage. And metaphase first stage. In metaphase stage, we also know that the metaphase plate will be formed. So all chromosomes are arranged on the equator of the chromosome. The chromosomes are arranged on the equator in a pair form. So there will be a double metaphase plate. So that is the difference between mitotic metaphase and meiotic metaphase. There will be a two metaphase plate and fuel. The two types of spindle fibers are formed in this stage, that is discontinuous spindle fiber that is present from pole to centromere, and the second type of spindle fiber is continuous spindle fiber that forms between the pole to pole. The third stage is anaphase first. In anaphase stage, each chromosome separates and moves towards the poles. The movement is due to the spindle fibers that pulls the chromosome on the pole side. So there will be a two type of movement seen, that is pulling movement. The second is pushing movement, that is due to fiber that is present. The last stage is telophase first. Telophase is also known as the reverse prophase. So in prophase stage, there will be a disappearance of nuclear membrane and nucleus. So there is a nuclear membrane and nucleolus began to reappear at this stage. So that is also called the reverse prophase centriole. So that is the process of karyokinesis. Now the second process is cytokinesis. In cytokinesis, there will be a division of cytoplasm. So cytokinesis occurs in animals by the process of fur formation, and in plants, the process of cytokinesis occurs by the process of cell plate formation. That is the complete process of meiosis first. Now the second process is meiosis second. So the process of meiosis second is similar to the process of the mitosis as we discussed earlier. So that is the process of mitosis. So there is a 14th chromosome in the humans. When the meiosis first occurs, meiosis first produces two cells, and that is the reductional division. So there will be a number of chromosomes, that is 23. So that is meiosis first. But in meiosis second also produces two cells, but meiosis second is a equatorial division. So there will be a no reduction a chromosome there produces 23 chromosomes. So the complete process of meiosis produces 4 cells, that is haploid cell. So the meiosis second produces 2 cells. And the process of meiosis are found in the germ cell. And the another process is amitosis. The term amitosis was given by the detail of the mitosis is given by them. So as we discussed, in mitosis and meiosis there will be a karyokinetic division and the second division is cytoplasmic division, but there is a no sequential changes like prophase, metaphase, anaphase and telophase. In a mitosis, division of cytoplasm and nucleus occur simultaneously by the constriction. In a mitosis, division may be equal or unequal. A mitosis is faster cell division which may complete in 20 to 30 minutes. Amitosis is cell division of prokaryotes. For the example, yeast reproduce by budding. That is a form of the amitosis.